Hi, good afternoon. These are exciting times. I'm going to show you how in the last 4 months, 8 months, we were able to improve the productivity of biomanufacturing by several orders of magnitude. Maybe here. So these 3 cell types are the factories that Biotech uses to manufacture the more complex and tiny machines known to man, proteins. These tiny factories crank out medicines, jet fuel, biomaterials, supplements for food, and in the future, far more. With our technology, we have tested all these factories, and we have measured unprecedented increases in productivity per hour. I'm having some issues with the... Where should I point? Sorry for this. The current method implemented today by the industry consists in large vessels with propellers in order to provide, optimal conditions for strain proliferation. Can you imagine the most complex machines known to man, the most sophisticated manufacturing pipeline, and we put it inside a blender? And in use, only for a brief period of time, all the conditions are just right for the intended reaction to happen. Before the peak, there's too few cells and too much media. And after the peak, there's too many cells and too little media. We were driven to develop a novel approach to stay at that magic moment in time, at the peak of the productivity curve. By designing the bioreactor around the cell itself, we came up with a completely different approach. Microchannels that sustain cells in a laminar flow, providing optimal conditions to every cell all the time. Gas exchange and mass exchange do not vary. We have functionalized this intricate lattice to be functional for biomanufacturing purposes. Here, you can see how using light and collagen, we were able to manufacture an artificial synthetic circulatory systems that we can program for the specific bioprocess that we want to pursue and stimulate the synthesis of protein and control the density of cells. We can also manufacture more complex designs in which we print 2 lattices that are intimately related. And by using a porous biomaterial, we can enable the diffusion of gas and nutrients, sustaining these cells in a continuous comfort zone. And as we zoom out, you can see our revolutionary design. Here, we are showing, for the first time in Argentina at least publicly, what we call a bubble-free bioreactor used in our bioprocessors. These tiny machines has an equivalent performance of a 1-liter bioreactor, but they are really small. They are so small that they can literally fit inside your pocket. And if we want to build a bigger bioreactor with more capacity, we just assemble a bunch of these together. So, remember this graph? This was our first goal. But what we found out is that this wasn't the upper limit at all. Here, working with E. coli for the production of human recombinant protein with one of our clients, we measure an increase of 70 times of increase in productivity over the baseline. The blue bars at the bottom confirms that we were actually manufacturing the protein in high densities every day of the process with a strain that normally would work for no more than 4 days. And these results are consistent with other work that we did. Here, working with other factories, HEC cells and CHO cells, we measure the same results. And these were with both adherent and non-adherent strains of CHO cells. On the other hand, media sterilization is imperative for biomanufacturing. We have developed another technology that doesn't rely on steam or high pressures in order to get media sterilization. Instead, we use electricity. What you're seeing here is a pulse of high voltage that bombards the media as it goes through the bioprocessor. This might look like a single pulse, but it's actually more than 150 micropulses. What this does is that it disrupts the membrane of undesired microbes. So, with devices that are 500 times smaller, we see a future of decentralized biomanufacturing. And to further enable this, we have designed a strain-on-a-chip method. Now, customers can pick a strain from a strain bank, and we will put that strain into a chip, and it's ready to go. They just plug it in into their Stämm and hit start. After 24 hours, they will get a continuous stream of growth culture, ready to inoculate their bioreactor. We are currently working with 5 companies, and we are intended to get to 15 by July. Our customers are benefit because of reduced capex, reduced time to market, and a really high decrease on OpEx as well. All the work presented here is protected. We have worked with 2 of the main law firms of Silicon Valley, and this is the first patent for a continuous laminar process of biomanufacturing that is both compatible with industrial research and scaling up and prototyping environments for the development of bioproducts. Federico and I have worked in this project for the last 6 years, and we have assembled an interdisciplinary team of leaders from bioinformatics to bioengineering. Each one of them is currently leading technology products within the company. And we have a seasoned team of advisors with decades of experience in the industry. They have taken products and tools through regulations approval, and they have articulated multi-million dollars with pharmaceutical corporations. We often heard that biotechnology holds the key for solving the biggest challenge that humanity is going to face in the next 30 years. But if biotech holds the keys, biomanufacturing is the room in which these solutions live. And we need more rooms because the main trend in the industry is to manufacture more keys. And if we want to put these solutions in the hands of the people all around the world, we need a different approach of how we think about biomanufacturing systems. So from Stämm, we invite you to be part of the next leap in biomanufacturing industry. Thank you very much.